This protocol provides a simple, rapid, and reproducible method to induce a scalable blunt force traumatic brain injury in adult zebrafish. The adult zebrafish that undergo this blunt force traumatic brain injury exhibit many of the characteristics observed in humans that suffer from a blunt force TBI. Demonstrating the procedure will be Mr. James Hentig and Ms. Kaylee Cloghessy, two doctoral students from my laboratory.
Begin by filling a petri dish with modeling clay, then use fingers or the back of a pair of forceps to create a raised platform with additional modeling clay. Divide the raised platform lengthwise into two approximately equal halves using a razor blade. Form the two halves into a channel such that it accommodates the length of an adult fish. Use additional clay to build walls to secure 2/3 of the fish body with the head exposed. Mold a small support in the exposed head region perpendicular to the walls to avoid rotation or recoil of the head upon injury. Ensure that the channel's sides do not impede the dropped weight. Use a mini hole punch to create a 3 mm steel disc from a 22 gauge steel flashing. Place an anesthetized fish, unresponsive to tail pinch, on the clay mold within the channel with its dorsal side up so that its body is secured on the sides. Place the 3 mm 22 gauge steel disc on the head centered over the desired impact point. Align the fish perpendicularly to avoid its head from tilting to one side, which could cause an uneven impact.